So hello, welcome everyone. Today we'll have Eugene talking about 3D structure prediction, and I believe how we can use 3D structure prediction to improve 2D structure prediction to improve 3D structure prediction. And yeah, I think it's a great way to celebrate end of vacations. So with that, so he finished his master's in applied mathematics at the Institute of Mathematical Problems in Biology, Pushchino in Russia in 2015. Then he joined to do his PhD in bioinformatics, the same institute under supervision of Mikhail Reutberg and Ivan Kulakovsky, and defended his thesis in 2021 at the Moscow Institute of Physics and Technology. His thesis was about the relationship between RNA structure and tertiary motifs. So to learn more about tertiary motifs, I believe he joined a lab of Janusz Bujnicki in Warsaw in 2021, and he's now ENBO postdoctoral fellow since 2022, and today we'll hear about secondary structure prediction, which is a very interesting combination of interest. Eugene, the screen is yours.
Thank you, Marcin. Hi, everyone. Yep. Once again, I'm Eugene. I'm a postdoc at Janusz Bujnicki lab at the International Institute of Molecular and Cell Biology in Warsaw, and today I want to present to you our new method of RNA secondary structure prediction, which is based on a really simple stem formation model. So as we know, non-coding RNAs are usually structured molecules that perform their functions by binding with other molecules, either with proteins or other RNAs or ligands or anything. So we can state that the structure defines its function, and thus we should study the structure better and understand how it's organized to be able to draw any conclusions about its functions. So we usually divide the architecture of RNA structure into 3 organizational levels, with the first level being primary structure, which is RNA sequence, being the string in the 4-letter alphabet. The secondary structure is a skeleton of its structure containing the, the set of Watson-Crick and Wobble base pairs, Watson-Crick GCAU and Wobble GU base pairs. Usually we depict secondary structure as a planar graph with edges being base pairs, and they form these short double helical regions that we call stems. Or alternatively, we depict it with arc diagrams or bracket diagrams where we stretch the sequence from left to right and depict base pairs as an arc or as a pair of matching brackets. And the third level will be a tertiary RNA structure, the spatial structure in 3D, where we include all the tertiary interactions formed on top of the secondary structure. So since, unlike for proteins, we don't have many structures known in the PDB databank, the problem of computational prediction of RNA structure, and in particular secondary structure, is still relevant. And again, unlike proteins, in RNA structure prediction, we have, in RNA secondary structure prediction, we have 2 major complications. The one is that RNA sequence can form not the defined single structure sometimes, but like alternative structures, as an example here. So here's the same sequence can form either 2 consecutive stems or hairpins, or an alternative fold with the pseudonode. And the pseudonode is another major complication. So the pseudonode is a thing where in bracket diagram, we have one part of a stem being intercalated between the parts of another stem. And when we have pseudonodes, the problem of the prediction becomes NP hard and polynomial algorithms don't work. So this is another major complication in RNA secondary structure prediction. So first of all, we formulate the problem as follows. So we have a sequence of RNA, of a single RNA, or a multiple alignment of homologous RNA sequences, and we want to predict the base pairs forming the secondary structure of the molecule. A little historical overview. So the first algorithm was developed in 1978. It was the Nasinov algorithm, and it was based on dynamic programming. And the problem was to maximize the number of base pairs in a structure. And it's depicted here on the left. So usually for educational purposes, we assign weights for base pairs. with GC base pair being the strongest because it contains 3 hydrogen bonds. So we assign a weight 3 to GC. We assign a weight 2 to AU pairs having 2 hydrogen bonds. And usually we assign weight 1 to GU pairs. They do have 2 hydrogen bonds, but they have a bit wrong geometry. So this is why it's assigned a weight 1. So for this sequence, the algorithm predicts like the maximum weight, the structure of the maximum weight of 8, including 3 base pairs. And that's pretty much it. So yeah, the algorithm does not allow pseudonodes because of dynamic programming cannot handle pseudonodes. Soon enough, we understood that RNA does not maximize the number of base pairs. And in the 90s, we developed, people developed another approach where we still use dynamic programming, but we don't maximize the number of base pairs, but rather we minimize the free energy of the structure. So this is on the right on the slide, an example. So we assign a free energy to a structure being the sum of free energies of its elements. So this loop includes 6 different elements. We sum up the measured somehow energies and obtain a free energy of the structure. And the task is to find the structure of the minimum free energy. And again, via dynamic programming approach. So again, we cannot handle pseudonodes here. And then a bit later, with an advent of more and more known 3D structures and secondary structures of RNA, some approaches started to use, instead of empirical thermodynamical parameters, statistical potentials derived from the known structures. And this group of algorithms we call to maximize the expected accuracy of the structure. Now we have a couple of variants of such approaches that can handle pseudonodes, but some of them still cannot handle the pseudonodes. And this is our current situation. So now we have even a number of deep learning based approaches as a spot RNA, for example, the diagram of which you can see on the right. So the methods are really, really complex, but the quality is still not so good. So on this benchmark from Spot RNA 2 paper, the left part is the alignment-based prediction, and the piece of the right part is the single sequence prediction. And we can see that surprisingly, they are on the same level more or less. So we cannot even predict better with an alignment rather than with a single sequence. And still the mean F scores here are below 80%, which is not quite good. So this is where we are currently. And now I will start from a year back with when we started coming to our new approach. So a year ago, we were solving a completely different problem in 3D. So we were trying to answer the question how we can superimpose 2 arbitrary fragments of RNA 3D structures. So an example of 2 arbitrary fragments you can see on the slide. And this is a perfect example of 2 visually similar fragments, but they actually differ in all the things we usually rely on. So they don't have the same sequence. So you can see that the bases are different. The geometry of the geometry of the interaction is also different. So the bottom base pair here has different geometry from the bottom base pair here, and the topology also differs. So we cannot rely on type of secondary structure loops or anything, because here on the left we see a continuous strand of 6 nucleotides, and in the right fragment we have 2 strands of 3 nucleotides. So how should we superimpose these 2 motifs? Actually, the computational formulation would be how we find the matching between the elements of these 2 sets. So between the nucleotides of the 2 structure fragments. So in general, the computational complexity of this problem is factorial, since we would need to try all the possible variants, and this is a factorial number. So we ended up with this beautiful idea. So let's assume that the perfect superposition of these 2 motifs exists. And then it's highly probable that at least 1 pair of nucleotides in the perfect superposition would be close to perfectly aligned. So would have a RMSD close to 0. And once we assume that such pair exists, we can just try the quadratic number of variants. So try all the possible single residue based superpositions of entire structures. For each of them, derive the subset of mutually closest bases, and then just choose the best variant, either the largest subset of mutually closest residues, or a subset with a less RMSD or with anything else what we want. And that's it. So with this beautiful idea, we now reduce the factorial complexity to quadratic complexity. And we were so amazed with this thing that we stopped and think what just happened? How did we do this? And what we did actually is that we just simply used the dependency in the data. So we don't have two sets of independent elements, but we have two rigid structures. So once we superimpose the entire structures based on a single residue-residue pair, we actually do have fixed, we did fix all the other elements, and we can derive information from it. This is what we did. So now we applied the same idea that actually that was discovered last year. We applied the same idea to RNA secondary structure prediction. So here for the two sets of elements, we would use the same RNA sequence. So here we have the same sequence on the top and on the left, and it would play the role of the two sets. The residues are the elements of the sets, and the base pairs would be the cells of this matrix. So we would, again, we'll solve the problem of partial assignment of the matrix between the elements of the two sets. So we will be predicting which matches will form base pairs. But since the same sequence plays the role of the both sets, the matrix will be symmetric. So we can forget about the lower triangle in this matrix. And since we know that RNA does not form hairpins shorter than 3 nucleotides, we can zero out 3 diagonals near the main diagonal. So this is this triangle is what we have left. Now we again here assign the weights of the base pairs, of the potential base pairs for GC, AU, and GU pairs, 3, 2, and 1. And now we were faced with a question like, if we assume that this guanosine, for example, forms first in the correct secondary structure, how it chooses the cytosine, it will be paired with out of 3 different options. And here we're answering the question, how can we use, what dependency in the data we can use? And easily and more or less obviously, we know that secondary structure don't have a lot of lone base pairs. So the majority, the vast majority of base pairs form stems. So maybe we can use this. So maybe the guanosine will form with that cytosine that is incorporated in the best stem, being best scored or the longest one, or how we will define it. And the stems on these matrices correspond to diagonals. So this diagonal of 2 GC pairs is a stem of length 2, and we assign for each base pair a weight, not the weight of the base pair, but the weight of its stem, of its entire stem. So this 2 6s is actually a sum of 3 plus 3, so we get 6. And now, more or less, we have less variance to choose. But now, actually, we have a problem that stems do overlap with each other. And since RNA actually don't fold in a moment, but it forms stem by stem, more or less, or base pair by base pair. So we choose to use a greedy approach when we first predict the best stem, and then in this case we would predict first the stem of 2 GC base pairs, and then we ignore, forbid all the inconsistent base pairs with the stem. So we zero out the rows and the columns of these two base pairs. So we get the matrix from the right, and now then the second best stem would be this diagonal of 3 5s. So this is the second stem that we will choose. So this is the approach, and that's it. Almost that's it. So this is the core idea of the approach, and this actually solves 2 again major problems that are known to be characteristic to current approaches. But first of all, the approaches that maximize the expected accuracy, they do this. So they assign probabilities or some scores to base pairs, and then they use dynamic programming to maximize the weight of the structure. And in the example here on the left, we would choose for sure to maximize the total weight of the structure. We would choose this stem of 5 base pairs with number 14, with the weight 14. But then to maximize the total weight, this 9 also will be selected because it does not overlap with these 14s. But thus we choose just a single base pair of a stem of weight 9, considering the score of the entire stem. And this is not exactly fair. So when we use rather a greedy approach, we choose this stem of 5 base pairs, we zero out rows and columns of this stem, and thus we kill this 9, and this 9 becomes a 3, and now we will not choose it if we don't choose long base pairs. So this is one thing. And another thing is that we now don't have any problems with pseudonodes. So on this slide, we have 2 different structures, both including 2 stems of each of which includes 3 base pairs, but only one of the structure is pseudonoded. So can anyone guess or tell like which structure here includes a pseudonode and which one does not? Any guesses? Okay, now it's over. Okay, so the pseudonode is on the left, but it's actually not obvious. So we actually needed to sit and think how we can identify where the pseudonode is. And we understood that, okay, we need to draw the lines towards the diagonals. And if they intersect, then this is a pseudonode. If they not, this is not a pseudonode. But actually, for a human eye, as well as for our algorithm, a pseudonode is not a special case anymore. So we solved one of the complications. It's good enough. But now we return back from 2D to 1D, since RNA still is a linear polymer. So what we do now is we predict stems one by one. So we annotate all possible stems, choose the best one, predict it, remove the inconsistent base pairs, and then iterate. So first we start with an empty secondary structure, a dotted structure here. So this is like the 2 best stems, the higher rank stems of the same BP score, so of the same sum of the base pair weights. And this one confines the shorter loop, and this one confines a really long loop. But since we know that in RNA long loops pretty much do not exist, we should prioritize somehow the stems confining shorter loops. And this we do this with a factor based on the distance. But still, of course, these stems do exist. The stems that span long regions of these sequences. But we will predict them only just later, after we did predict the things within, like the more close stems, and thus we will consider only the distance between the inner stems when we will predict this long stem. Another thing is that we know that pseudonodes of higher order do not exist, and the order of a pseudonode is like the number of different bracket types that are required to depict it on bracket diagram. So we also penalize like stems that would form a higher order pseudonodes. So this stem, for example, intercalates with a single stem here, and this one intercalates with the stems of 2 different levels. So we will penalize this stem more and this one less. So we will choose this one. So this is the second factor. And the third one is simply that we know that some loops are more favorable than others. So we have bonuses for generate tetra loops and for short symmetric and close to be symmetric internal loops, like the internal loops here, So this is the entire formula. That's it. And the last thing would be that, of course, we would face situations where we have equally good stems and and we have a suboptimality threshold of like which within the threshold we consider stems to be equally good. And in these situations, we simply bifurcate and add all the stems from our within our threshold, but separately, and we continue predictions separately for each variant. And it results in a number of alternatively predicted structures, which we afterwards, either from a single run or from several runs with different parameters. And afterwards, we rank the predicted structure based on some on structuredness. And if we have a reference structure, as we have here in green, we would measure the quality in defining the true positives as a number of correctly predicted base pairs, the false positives as number of wrongly predicted base pairs, and false negative as wrongly missed base pairs. And we use F-score as a target metric, and we completely avoid defining true negatives because it's not quite obvious how to define true negative, so we've chosen to avoid it. Now that's it for sure, and this is the first results of our benchmarks. So what here the word means, so different words mean. So we selected a data set of 274 sequences with the secondary structure from PDB data bank, with no chain breaks, with no missing nucleotides, with no modified bases, like really great data set, and a non-redundant one. Then we train the parameters, and we're choosing two different configs of SQUARNA. The first one, the default one, relies on two different parameter sets, and the structures being ranked afterwards together, and another one with a single best parameter set. This is an alternative configuration. And since we do predict alternative structures, we consider two different cases when we consider only the top one structure or the best of the top 5 structures. So what we see here, and this lines shows the groups of statistically significant differences. So we don't have the significant differences within the groups, but do have between the groups. So what we see here is that if we consider our top 1 predicted structure, we are on par with the conventional tools like RNA fold type nodes and the shed nodes. And if we consider the best of the top 5 structures, we are on par with the best deep learning methods like MXFold2 and SpotRNA. And it's like why we choose the top 5 and not top 10 or anything, because the CASP and RNA puzzles contest allow up to 5 submits to each target, so it's a good number to choose. So it's not exactly fair to compare. Well, first of all, it's not exactly fair to compare our 5 structures to just a single structure from deep learning, but still the number is not too high, so we did it. Yeah, and it's not also quite fair to compare performance on the data set on which we trained the parameters, although we do have like 10 parameters or so we cannot overfit to anything, but still we also did the performance comparisons on the TS1 dataset from the SpotRNA paper. And here we have only 62 sequences, so there is no statistically significant difference anywhere, but the overall picture is the same. So the group of 5 looks the same on the left, and the group of 4 looks the same on the right. So to conclude, with a single sequence approach, we could achieve the level of the best methods currently existing with a much simpler approach, and this is good enough. Yeah, and once two notes here. So first of all, the overall performance of all the tools a bit worse for TS1 dataset than for our dataset, and this is because TS1 dataset has a number of things that actually like of bugs of some kind. So this is one of them. So here, this sequence, this is actually a fragment of a sequence. Here we have a 0 length hairpin, and we know that such things do not exist in RNA, and this is simply a result of artificial deletion of the missing hairpin from PDB. So actually, deep learning methods do not care about such stuff, but we do. So this is why on the TS1 dataset, all the tools except deep learning ones perform a bit worse. And
Eugene,
also, yeah.
can you, what's the source of this data set? I missed that. This TS1?
TS1 is spotted from SpotRNA paper.
Oh, okay.
Yeah, so it's TS1 reduced. So the TS1 dataset includes 67 sequences, but actually they do mention in methods that they use TS1 reduced, which includes only 62 of them, because they throw out the 5 sequences with modified bases, because some of the methods cannot handle modified bases. But yeah, it's from the SpotRNA paper. Yeah, and the second thing that we spotted that some of the methods perform slightly worse for longer sequences than for shorter ones. And this is the case for us. First of all, because like we do use faster but of worse quality configs for longer sequences because of the time complexity. So we cannot run good configs for like ribosomal RNAs, since it would take ages to finish. So this is why we do have 85% of mean F score for SR trained dataset, but also we calculated to check this differences between longer and shorter preferences, shorter sequence preferences. We also calculated total F scores, and this is when we sum up true positives, false positives, and false negative over the entire dataset, and only then we calculate the total F score. So in the total F score, longer sequences contribute much more, and in mean F score, all the sequences contribute equally, which is not fair, both ways not fair. So we also calculated harmonized F score, which is the harmonic mean of the two. on the total end of the mean. And having these three different F-scores, we can see like if mean F-score is much higher than the total one, then we can understand that this method prefers shorter sequences much more than the longer ones. So this is just a note.
Eugene, here as well, I think for RNA fold, you pick only one sequence, yes?
For RNA fold, yeah.
Yeah, like you don't use top 5 sequences.
Do RNA fold predict alternative structures?
I think, yeah. Always.
I don't think it predicts the minimum free energy structure. I don't think it's. I could use RNA support actually, yeah, but I didn't. But yeah, this is a good observation. Okay. Yeah, thank you. Okay, now moving to an interesting part. So now the alignment-based algorithm. So the algorithm that we use basically relies on the same ideas. So here on this example, we have 3 sequences in the alignment. We do calculate STEM BP score matrices for each of the sequences, and then we sum them up to obtain the total STEM BP score matrix. The problem here is that we don't have a definition of a STEM for an alignment. We only have the STEM definition for a sequence because of the gaps. So here for each individual sequences, we first remove the gaps, calculate the STEM BP score matrix, and then realign it, adding 0 columns and rows in the gap positions. And then the realigned matrices are being summed up to obtain the total matrix. And here again, since here the base pairs of the given stem can have different scores now, which was not the case for single sequence. Here we predict base pair by base pair and not stem by stem. So in this case, we will choose 5 cells with number 21. And after we choose this set of like the best base pairs, we use it for a second iteration as restraints. So we selected these 5 base pairs. We used it as restraints on the second iteration. So we removed all the inconsistent base pairs. And then we again decalculated the same matrix. And we did the same thing again. We selected these 5 base pairs again. So why we need the second iteration is because we can have a case of 2 conflicting stems of the comparable score. So we can choose like some of the base pairs of one stem and some of the base pairs of another stem, and we actually wanted to kill each other and to zero out from the final result. So this is why we use 2 iterations. And on the step, we also either apart from 2 iterations of step 1, we also have step 2 when we use the total stem BP score matrix to guide the single sequence predictions of the member sequences, from which we derive the consensus, and this is the step 2 predicted structure. And then having 2 structures, we on the step 3 we return either the intersection of the 2 or the union of the 2. So this is for settings that we used and that we used in the benchmarks. And this is again, that's it. That's the whole approach. So we did train a couple of parameters on the short data set of 11 families. This is, we called it RNA Starline Next data set. So it includes 8 families from RNA Starline data set, which is often used for these purposes. But we also added 3 underscored families here, having well-known pseudonode structures just to have more pseudonodes. So the depth of the alignment here is defined as simply the number of sequences. The length of the alignment is the length of the alignments of all the sequences with all the gaps. So the distribution of lengths here is quite good. So we have lengths of 2000, of 1000 to 100. And the distribution of depth, so we have 6 families of really deep alignments, 1 family of just 12 sequences, and 2 families of 33 and 37 sequences. So this is the data set that we trained on, and this is the results that we obtained. So we used Centroid alifold, RNA alifold, IPNOT, and RScape, or CACOfold. And we used for RScape, we used 2 different settings where we have only the nested structure from the first line of the output, or a total structure where we have the nested one with an addition of all possible base pairs that would result in pseudonodes. So first of all, we obtained this. So we see that SQUARNA is the only tool that produces the medians and actually as well as means higher than 80%, unlike other tools. But actually here we were puzzled with this performance of RScape, And soon we understood that the reason for this is just that RScape requires a lot of sequences in the alignment to predict secondary structure confidently. So we selected the 6 families with more than 300 sequences within the alignment. And here RScape outperforms the conventional tools, but still SQUARNA is better than even RScape. So we were quite happy with it, but of course this is too small data set to compare it, to draw any conclusions. So we actually selected the entire RFAM data set, and this is what we got. So we obtained the data set of more than 4,000 families, and we did this graph where we have on x-axis the threshold on the alignment depths. So this is on the left here. This is the results for these tools based on all the alignments, all the seed alignments of the families, and moving to the right, we remove the alignments of less depth. So on the right, we have only really deep alignments. So what we can see here is that on deep alignments, SQUARNA outperforms everything. RScape outperforms the dotted lines here. But on the left part, something strange is happening. So we have this really huge gap between the centroidal fold and RNA fold, and SQUARNA and RScape. But seeing this tails, this unusually high tails of RNA fold and RScape, we actually hypothesized that for shallow alignments of like 2, 3, and 4 sequences, in RFAM we have not confidence secondary structure, but predicted secondary structure. And it just people often use either RNA fold or RScape to predict secondary structure. So this is why we see these tails. So actually we don't compare ourselves to the true structure, but to the predicted structure from the 2 tools. So then we selected a subset of families with at least one 3D structure in PDB. And now we don't have the gap anymore. Yeah, we have 134 families of such dataset. So now we don't have the gap, but the overall picture is the same. But something strange happens here. So since this is in harmonized F-scores, so here longer sequences contribute more. And we actually spotted that long ribosomal RNAs that are hard to predict, they're represented in seed alignments with an alignments of about 100 sequences. So this is an effect of ribosomal RNAs. Once we remove them, we become better. Once we don't remove them, we become worse. So we needed a cleaner data set so we actually, what we see here is an effect of unequal distribution of depths among the RFAM families, or the RFAM families among depths. So different depths, we have different families, and this is the thing that precludes us to see the pure depth effect. So we actually did the following. For the 11 families of RNA Straline X dataset, we subsampled alignments of a given depth. So for each family, we sampled 10 alignments of 2 sequences randomly, 10 alignments of 5 sequences, and so on until 300 or until the seed alignment depths allowed us. So some families end up not on the right. So each dot here is a prediction, is an F score of a single prediction. Solid lines are the mean F scores out of 10 alignments for each family, and the dashed gray line is the mean F score among the families. So this is the result for centroidal fold predictions, and we can see that it does increase from 2 sequences to 5 sequences, but since then it actually stabilizes between 0.7 and 0.8 and does not grow anymore. So centroidal fold does not care whether we have 5 sequences or 300 sequences, it will produce the same result. So we assume that it predicts like somehow minimum free energy structure, and uses covariation only to reduce the variance of the prediction, but not to guide actually the prediction itself. So we were surprised with this, and then we obtained the same results for RScape. Here, thank God, we have the increase with the depths. That's good enough. So we have highly confident predictions on the once we have 100 sequences and more. But actually, before 100 sequences, the results are pretty much unstable. And for example, on 10 sequences, the mean F score is below 50%, which is not good enough. So it's on 10 sequences, we don't have enough signal for RScape to perform confident prediction. And this is what we see on SQUARNA, on one of the settings of SQUARNA being step 3U, but actually for all the 4 settings, the results are pretty much the same. So we are stable from like 20 sequences or 10 sequences, as you define it, we reach 90% of mean F score around 100 sequences, but already on 10 sequences, we have mean F score of 80%, which is like the beautiful picture. This is what we want to expect from, what we want to see from the alignment-based prediction tool. So this is a cumulative graph. The four solid lines are the four SQUARNA settings, so they are pretty much stable. We have IP node here, which actually decreases after 30 sequences, and we don't know why. And we have RScape, for which like enough signal starts to be happening only on 75 sequences. And actually, this pretty much solves the main problem, since in ARPAM we don't have much deep alignments. So only 8% of the alignments have more than 50 sequences, and about almost half of the alignments include from 5 to 50 sequences. And this is where SQUARNA outperforms everything. And again, more than half alignments have from 2 to 4 sequences. So for this part of our PAM, and this is pretty substantial part of 46%, SQUARNA beats everything. So this is quite good, and we were quite happy with it. So of course we checked also the effect of sequence similarity, since on the same depths, on the same number of sequences, the prediction quality should depend on how diverse your sequences are. So on the 5 identical sequences, you don't have actually the 5 sequences, you have the same sequence. So we again subsampled different alignments for depths 5, 10, and 20, and plotted the Pearson correlation coefficient between the prediction quality and the mean pairwise sequence similarity within the alignment. So actually we expected to see negative correlation here, because like the less the similarity, the more diverse the sequences, the more single signal we have, the better quality of the prediction we should obtain. And this is what we see for SQUARNA. So indeed we have not much, but not high, but clear negative correlation, and it diminishes with depth. So with the growing number of sequences, we're starting to get enough sequences, and we started to be able to ignore how diverse they are. But for other tools, we don't see a correlation at all. And for RScape, it's not surprising, since RScape on this depth is not stable, so we can see anything here. But for the RNA alifold and centroid alifold, we were surprised to see even like close to zero correlation, or even a positive correlation. Then we looked into
maybe another question, because here you talk about alignments, but I think you talk about seed alignments, yes, only.
Yeah.
Yes, because then you have full alignments, and so then you have hundreds of thousands sequences. Yes,
that's true. Yeah. But here it's not even seed alignments, it's sub alignments of the given depths.
Mm-hmm. Yeah. But even before, in other cases,
initially, we use
like more sequence, like you can get more sequence using the alignments, making models, and then fetching more sequences, so then you may have deeper alignments.
Yeah, that's true. Yeah, so it's another question of what quality the alignment will be, but yeah, you can. Okay, so then we analyzed a couple of fails of SQUARNA on deep alignments. So this is one of the examples. RFAM family 2929. It includes almost 500 sequences. We have a single stem annotated in the RFAM structure. SQUARNA predicts an alternative pseudonode, and RScape predicts the correct stem along with the second stem. So here we analyzed the sequences, and we understood that we have only 90 unique sequences here, and the 3' half of the entire alignment shows unusually high sequence conservation. So it's like the mean sequence similarity on the 5' half is about like 0.6, 0.7, and of the 3' half, it's about like 0.95. So we assume that we have some sequence motif there. This actually misleads SQUARNA from the correct stem to seems to be false pseudonode. But still, in the verbose output of SQUARNA predictions, we can see the correct stem being the third more conserved stem. And in the individual sequence predictions, we will see alternating faults of the correct stem and incorrect pseudonode. And once we actually try to subsample and select like 5 the most diverse sequences, 10 the most diverse sequences, 20 and 40 and 80, we see actually that until the depth of 40 sequences, SQUARNA predicts the same the RScape does. So actually, SQUARNA can be misled by sequence conservation over covariation, which RScape is immune to. But still, you can manually curate your alignment and it will help SQUARNA to fix the case. So this was a bad example. This is a good example. This is a sequence of this is an alignment of S15 ribosomal protein leader sequence of RFAM family number 114. So here it's known to fold into two alternating structures. One of them includes 3 hairpins, 2 of which are annotated in the RFAM structure, and the alternating fold is a pseudonode. And here SQUARNA predicts the correct pseudonode, RSK predicts just the first hairpin, and epinode predicts the 2 correct hairpins. So SQUARNA here is the only tool that can predict the pseudonode. And again, in the verbose output, you can see like the first hairpin, the first hairpin's base pairs, the base pairs of the pseudonode stem, and here we have 3 base pairs of the second hairpin. So we can conclude the alternating folds. And also in the individual sequence predictions, you can see also the alternating folds. So for some sequences, one structure is predicted for another sequences, another structure is predicted. So you can analyze the verbose output and understand what's happening. And last but not least, we also made SQUARNA to be able to incorporate shape reactivity input data. And for this, we used the dataset from ShapeNotes paper of 24 sequences with the shape reactivities. And so we prepared the dataset in 4 different settings with a single sequence input only, single sequence plus shape, alignment, and alignment plus shape. And for this purpose, we built the alignments in completely structure-unaware manner using BLASTn to derive homologs, and then morphed to align the sequences based on purely the sequences. So surprisingly, at least for me, the alignment-based predictions in general, much worse than the single sequence predictions. So I would expect that they won't be better, since we know that sequence-based alignments are not good for RNAs, but they're much worse, and I would not expect that they would be much worse, but this is a good thing. So if you have an alignment of poor quality, you actually should use ipNOT and not the good tools. But still, when we wanted to understand how well SQUARNA can incorporate shape data, and we can see that with shape, it performs about 10% better than without shape for single sequence, and about 2 or 4% better than without shape on the alignment-based predictions. And actually, for sequences under 200 nucleotides, SQUARNA, out of the top 5 structures, SQUARNA predicts better even than ShapeNotes. And on this data set, which is like the data set on the part of which ShapeNotes was trained also on, and we also trained on, but we trained like at 2 or 3 parameters. Yeah, so we also, apart from shape reactivities, we can also incorporate structure restraints and reference structure, and we can handle multi-chain input. So overall, out of 10 features of the tools that are listed here, SQUARNA can do 8 of them, and the second best is ShapeNotes, listing just 5. So we cannot handle circular molecules for now, but we will soon add them. And we cannot handle, and we will not be able to handle non-canonical base pairs because they are simply not a part of secondary structure. So and we don't use deep learning methods. So that's it. So to summarize, so we presented here like a simplistic approach for RNA secondary structure predictions that is on par with best methods on the single sequence, and outperforms the alignment input tools, especially on the alignments of not enough sequences. And we developed the tool to be able to incorporate a lot of additional input data. So here I want to thank people involved in this project. So David Bogdan, who did a lot of technical dataset cleaning and other technical stuff with the data. Grigory Nikolaev, who now prepares the C++ version of SQUARNA. So now we implemented SQUARNA first in Python. Now we will have also, thanks to Grigory, C++ versions that would be much faster. And of course, Janusz Bonitsky for the overall supervision and for tons of the practical world examples. So we did develop the tool not in vacuum, but arranged on the practical examples. So this is a link to GitHub. Yeah, the work was supported by National Science Center of Poland and by the grant of National Science Center and by the fellowship of EMBO. And here you can see the link to GitHub. And within a couple of days, we will have the link to bioRxiv in the repository description. So we submitted the bioRxiv preprint, but it's not out yet. So this is it. Thank you. I would be happy to answer your questions if you have them.
Okay, thank you very much. So yeah, one of my questions was about the speed, but yeah, since I think you consider rewrite into C++, so probably it was not the fastest in Python.
Yeah, it's actually for sequences under 200 nucleotides, it's close to be the fastest. Of course, the fastest is RNA fold, but all the other tools are quite slower. So when we are on par with them on the speed also. But for alignment-based predictions, actually the SQUARNA step 1 is purely quadratic in time, and it runs like as really quick. Again, we're here the second best after RNA fold, but still it's really fast. So if you want a fast alignment, a good fast alignment based tool, you can use SQUARNA step 1.
Okay, more questions?
I have a question about, I guess, your comments or whether you've compared against Spot RNA 2, which, is kind of deep learning plus some evolutionary data.
Yeah, we did not compare ourselves to Spot RNA 2 because it takes a single sequence as an input and then builds the alignment on the fly. So it's not obvious how to compare to which tools we should compare it. So it's not fair to compare to single sequences since it does build the alignments, and it's not fair to compare to alignment-based sequences since it has a single sequence. So I could not find the option how to use an alignment as input. So if the authors of Spot RNA 2 will see possibly this record, please tell me how to use the alignment to disable the step of building the alignment and to use the alignment purely as input. Yeah, but for now we just don't know how to incorporate it in the benchmarks.
Okay, there is a comment and question from Sergey.
Yeah, I see the question in the chat. So I don't know if you're aware, the quadratic algorithms have been used in protein structural alignment. A good example is KPEX by Dave Ritchie. No, unfortunately, I'm not a protein guy, so I don't know a thing about proteins. It's good that they have also quadratic algorithms. And the question, what is the quality of RNA multiple sequence alignments? So in at least for the subset of families with known 3D structure, the quality is quite good. So the alignments, the seed alignments of this 134 families, this is good, and we can orient on this. But in general, for the entire set of more than 4,000 families, it actually differs. Some alignments are good, but some alignments are not quite good. And even from in the dataset from ShapeNote's paper, the shortest sequence is PRECU1 riboswitch, or ribozyme, PRECU1 ribozyme, I think. It includes a pseudoknot, and actually the pseudoknotted part of the structure is misaligned even in the seed alignment of R-Found. So this was actually also surprising. So actually this differs. And the worse, the alignments are much worse for the pseudoknotted stems than for the nested ones. This is for sure.
Yeah, I have a question here. So first, it's very nice to see non-deep learning methods do so well. One quick question is about, you said the R family, the over 4,000 families without, I mean, there are most of them without 3D-dimensional structure. So what's the ground truth of the secondary structure? I might have missed that.
Yeah, so again, it differs. So if we have really well-studied families that we studied for 30 years, and we did a lot of experiments other than 3D structure determination, we can we assembled a lot of data. We can more or less confidently assume it's secondary structure. But in some other cases, the secondary structure is just the secondary structure annotated in our farm is just a prediction of some alignment-based tool, and it's not always the correct structure. But in general, I cannot assess the mean quality of can we believe the secondary structure annotated there or not. But we can believe it only we can believe it fully only once we have the 3D structure now.
I see. Can I ask a follow-up? So
Yep.
So I wonder whether you looked into you compared it with some of the deep learning methods like MX-fold2 and spot RNA. Also, there's AlphaFold derived deep learning methods for RNA. I think they mean to predict a three-dimensional structure, but they also output the secondary structures. I think they do claim very good performances on the secondary level. I think the most recent one, called UniRNA, they at least on the paper they report nearly 0.9 or 90, even over 90% in terms of the, I think, either F score. So have you looked into the comparison with those kind of deep learning architectures?
Yeah, unfortunately, no. So at least I didn't hear about UniRNA. So we mostly focused on the benchmarks from the Spot RNA2 paper from 2022. And so we used the best tools from those benchmarks. But yeah, I should check the UniRNA, and maybe we will incorporate it if it's good enough. Thank you.
Oh, thank you.
I have a question. So you describe it as a modification to the algorithm of the dynamic programming algorithm. Can you instead modify write it down as a modification of the energy function, and then use normal dynamic programming algorithm? What I'm pointing to is normally, for example, from Vienna RNA, you get much more than the minimum free energy structure. You also get suboptimal structures or the partition function. Can you also get this from SQUARNA?
Yeah, from SQUARNA, actually, I do have the formula here. Yeah, so this is, we return a number of alternative structures, and we do rank them according either to structuredness, which is a structure score, here, or if we have the reactivity input, reactivities input, we also can rank them based on the reactivities or the product of the two scores. So now we use for the ranking, we use this simple formula. So when this power is set to 1, this is simply the sum of the base pair scores, base pair weights. But actually, after the training procedures, we set this power to 1.7. So we did prioritize longer stems over the shorter ones, but still this already works quite well. So the quality of the top 5 structure only like 2% worse than the quality of when we consider top all structures. So when we consider all the like hundreds of structures, we will get only 2% better F score than when we consider only top 5. So this is already a good score for us. And but of course, the beauty is that the algorithm is simplistic, so it can be extended in different ways. So now we use only base pair weights. We can extend it to base pair pairs weights for stacks, as it's done actually in minimum free energy free energy minimization. We can use sequence specificity. We can use ranking based on free energy, on different assessments of energies or different scores. So this is just a background that can be extended in many ways. But yeah, we didn't use, this is a good thing bad thing that we didn't use RNA subopt. I actually know about RNA subopt, but since it was not present in many benchmarks, I just simply forgot that we can use it and compare it to ourselves more in a more fair way. So we will for sure add the benchmarks to RNA subopt and maybe some other tools that do predict alternative structures.
Just a follow-up question. So if I have a sequence and a structure, can I use your approach to calculate the free energy or score of this specific structure for this specific sequence? Or it is only that I can start from a sequence and I will get the predicted structure? Yeah,
you can start with the sequence and with this known structure as restraints. And if this known structure is good enough, the predictions would be close to identical to your structure. But no, the fair answer is, the honest answer would be no. So we used just we cannot evaluate yet, taking a sequence in the structure, evaluate the score. But actually, since this formula is that simple, actually anyone can do it by themselves. Knowing the structure and the sequence, it's just this simple formula as we compute the score of the structure. But yeah, it's a good idea. We will add the evaluation mode. Thank you.
Thank you.
No, we have
Right.
more
Is there
questions.
any more questions? We've reached the hour mark, so I think if there's any more no more questions, we'll call it a day. The video will be uploaded in a couple days, so for any of your colleagues that missed the talk, they can watch it in our playlist on YouTube. And we will see you all in 2 weeks' time.
Thank you again for the speaker.
And thanks to organizers for having me here.